welcome to this screencast where we're going to be looking at our PowerPoint on the key knowledge 4 point regarding gene regulation. In this, we're going to be looking at this part of the study design. So it's the basic elements of gene regulation with specifically the prokaryotic Tryptophan as a simplified example of a regulatory process. So we're going to be looking at the Tryptophan towards the end as a specific example of how genes can be regulated. First and foremost, what is gene regulation? What do we mean when we say this? Gene regulation is where we actually regulate and control how much, when, and where a gene is expressed. And when we say a gene being expressed, we're referring to that gene's protein product actually getting made. So if we're regulating a gene, we're controlling when, where, and how much of that gene is gene's product is actually created. So generally that's protein being synthesized. And genes are really tightly regulated so that only the right amount of their product is made, only the right cells make it, and that they only make it at the right times. And it's really important because you don't want cells expressing genes that they're not meant to, or expressing too much and wasting resources and energy, or just not expressing enough and actually causing other issues for the cell. In terms of when genes can be regulated, you can pretty much regulate the expression of a gene at any point between you starting with DNA and having a completely folded protein at the end. So you can see in the diagram there, you could regulate the gene being expressed by inhibiting transcriptions, one of the first places you can stop things. Or if transcriptions does occur, you could stop the processing of the RNA into mature mRNA. Or even if that happens, you can have it so that the mRNA gets degraded quickly when it's trying to be exported and get to a ribosome. And that would stop it from continuing on. Or it can get to the ribosome, but translation at the ribosome is inhibited. You can even get to the point where your original instructions with the gene have been transcribed, they've been properly processed, they've been translated at a ribosome, yet you stop the proper folding of the amino acid chain into the final protein product. And you could even get to the point where you have a protein, but you have other things present that break that down before it does anything. So you can pretty much control gene regulation at any point you need to. Generally speaking, the main form of gene regulation and the main place genes are regulated are back at the transcription stage. That's the point that we're mainly focused on. It's just worth being aware that genes can really be regulated at any point in this process. But transcription is the most common and the one that we will be looking at. Now, we've mentioned types of genes before. I just want to come back to this because it's really important to be clear on the different types of genes, particularly structural genes and regulatory genes. There's a whole range of different genes out there based on what they might make or what they do and how they interact with other things. In eukaryotes, there's lots and lots of these different types of genes that play lots and lots of different roles. Prokaryotes are much simpler organisms, so they have a much more limited range in terms of the types of genes they have. So now, some of the key types of genes, we've got 4 listed here. So we have these constitutive genes. Now, these are what you might call housekeeping genes. They're the kind that are basically always switched on. They're always being transcribed. And that's because the things that they make and produce are going to be required by all cells all the time. Those sorts of things. The next kind of gene is structural genes. Structural genes code for proteins. They might code for RNA fragments that actually are not used in gene regulation. So something like rRNA, which is ribosomal RNA, they might code for that. They could code for a protein that might be structural or a protein that might have a specific function. And it's important to be clear that structural genes, at the end of the day, we would say that they code for a protein product in some way that is going to be used either as a structural element contributing back to the cell/organism, or it is a functional protein, that it's performing some specific function for the cell/organism. So enzymes, protein channels, protein components. The other option we said there was that they might make RNA that's not involved in gene regulation, so rRNA would be an example of that. One thing that they never do is that they never can code for a regulatory factor. So they will never make a regulatory protein. That is the domain of regulatory genes. Regulatory genes make transcription factors. Regulatory genes, they make the things that regulate and control other genes. So regulatory genes make transcription factors, which are proteins that help control transcription and regulate gene expression. What transcription factors do, and we learn about these again a bit later, is they will bind to DNA sequences close to a section called the promoter, and you generally have two types of transcription factors, either one that's an enhancer or one that's a repressor or an activator. Sometimes the enhancers are called. So regulatory genes produce things that will regulate and control other genes. The genes that they regulate and control are our last category there, regulated genes. These are ones controlled by the transcription factors regulatory genes make. What that means is your regulated genes can change how they're expressed, because their expression is dependent on how they are being regulated. So if there is a transcription factor present or if it's absent, that will have a difference in effect on how that gene is being expressed. So regulated genes are ones that can change up based on transcription factors, how they are expressing themselves. So those are four key categories of genes. Structural genes and regulatory genes are probably the two you'll hear talked about the most, and the two that you have to be very clear on what they are and their differences. So taking this, we have an exam question here. It says, describe the functional difference between a structural gene and a regulatory gene. So you can pause the video and answer this. We will have a video with the answer to this, an explanation of it, in a video of its own, and that will be linked in the description. So you pause the video, go and answer this, and then go and check your answer in the answer video. So you can do that now. So why, though, do genes need to be regulated? Why all this talk about regulation? I want you to have a think about it. If every cell in our body contains all our DNA, because you have your set of DNA and every single cell has a copy of that in its nucleus, that DNA contains all your genes. If we don't regulate how those genes are expressed and when they're expressed and how much they're expressed, what we're talking about is having those genes be active in every cell all the time. What would that mean? What would happen if that was the case? What would happen is that we'd end up with cells expressing things that they don't need to express. At times they don't need to express it. You don't need the cells in your liver to express genes for the cells in your skin. That's just not needed. So it would be wasteful for them to do it. It wastes energy and resources in the cell to do that if they're not needing it. And it would mean that we don't have our specialized cells, that they're trying to do too much. So to make sure we don't waste energy and resources and our cells only do what they need to do, we essentially either turn a gene on or we turn it off. So a gene that is turned on is one that its protein product or whatever it codes for, that thing is being made. A gene that is switched off is not doing that. So genes are turned off and on in different patterns, in different cell types throughout development. Okay, it's what means your cells look and behave differently from each other. Cells that are the same kind and have to do the same job generally will express the same genes in the same way at the same times. But when they're in different areas or have different roles, they're going to do that differently. You're going to have some genes that are active for your entire life, particularly anything that's involved in, production of enzymes for cellular respiration and that sort of thing, because all your cells need to be able to do that, and they need to be able to do it all the time. You need those genes being expressed all the time in every cell. Some genes will only be active for a certain period of time. the genes that you might need at the start of puberty to have expressed to produce hormones, you might not need them to continue to be expressed in the same way or at all. So they'd get switched off, and they don't need to be produced before puberty. Depending on the cell you're talking about, there are going to be genes that just don't need to ever be switched on because they've got no relevance to what that cell does. So only those genes that actually contribute to the cell being able to do the function it's meant to do are going to be switched on. Sometimes you get genes that are not switched on until well into adulthood. So there can be real variance with whether a gene is switched on or off, and it's for a range of different reasons. So now we're going to have another go at an exam question. You can see here it says 2 different cells taken from the same human were viewed using a microscope. The diagrams below show the structure of the 2 cells not drawn to the same scale. And you have a neutrophil, which is an immune type cell, and a neuron, which is a cell part of a nerve. Which one of the following is a correct conclusion to reach when comparing the 2 cells? A. At any given time, the genes expressed in each cell may be different. B. All proteins in each cell will have similar tertiary structures. That means similar 3D structures. C. The 2 cells have the same proteome. Or D. The 2 cells have different genomes. So you can pause the video and take a moment to think about your answer for that. Again, there will be an answer video for this question going over the answer, which option is right and why the other ones are wrong. And the video for that will be linked in the description. So you can pause the video and go and have a think about that question now. Now, there are other benefits to gene regulation. We've talked about sort of the basics of what it is, but some of the good things about it is when you can regulate genes and switch them on and off, it means that cells are not locked into behaving in just one way with their environment. When their environment changes, they too can change what genes are being expressed. And if the environment changes and suddenly they don't need to express a gene anymore, they have the ability to switch it off. And the reverse is true. If the environment changes and suddenly they need to express a gene, they will have the ability to switch that on. Now, this is all within reason. And, if they don't have the gene that's going to help them with the change in their environment anyway, then it's irrelevant whether or not it gets switched on and off. We're just talking about changes in the environment where there is a gene that is connected to that change and has some interaction with whatever has changed. So if there's a massive change in the environment that you don't have a gene that can respond to that in any way, then you're not going to see a change with that necessarily. So just be aware of that. This is only about genes that actually connect and can respond to that specific change in the environment. Something else to remember is that, like I said earlier, gene regulation can occur at heaps of different points, but most commonly that's at the level of transcription. So for the most part, if you're controlling a gene and you say switch it off, what you're talking about is you're saying that the gene won't be transcribed at all. And when you talk about sort of switching on genes, we're generally talking about promoting transcription happening. And this is a good thing if we're controlling things back here, because you're really controlling a lot of the energy and resources that are going to be needed in this process. If you stop transcription happening, you don't waste any resources even early on and that sort of thing. So controlling that level of transcription means that energy and resources are conserved and used only for what they're actually needed for, and they're not wasted on other things. So this whole process of controlling is also able to determine and dictate the amount of the protein product that actually gets made by the gene at any given time, so that you're only making what you actually need for the cell. Now, we've sort of danced around bits of regulation and some terms, and we talked about transcription factors before. I want to address what they are in a little bit more detail. They are proteins, and their job is to either switch on or off genes. And the way they do that is they attach and bind to DNA near the gene in question. There are a couple of different types of transcription factors. Activators are going to boost transcription, and repressors decrease transcription. They do what their name implies. Activators help activate and boost that transcription. Repressors help stop and repress transcription. Where they attach to the DNA are specific regions themselves. They're not part of the actual coding section of DNA, but they are nearby to that. So if you want to enhance and boost transcription, you're probably going to have an activator attached to a site called an enhancer site. Whereas if you want to stop something being transcribed, you're going to have a repressor attached to a region called a silencer. And so these are two areas where these transcription factors can physically attach to the DNA so that they can interact with molecules like RNA polymerase and either boost what they are doing or stop them from doing what they would normally do. What you have here is transcription factors allowing cells to do what we'd call logic operations. Logic operations are that sort of thing where you're saying, if this thing is true, then do X, Y, and Z, whatever this other thing is. Okay, and that's your logic operation. And so cells are able to go, oh, I have an activator, and therefore we will transcribe, or I have a repressor, therefore I will not transcribe. And so you can see in the image down the bottom on the left, you have an activator that has bound to its binding site. And what it does is it interacts with the RNA polymerase and it helps boost and promote that RNA polymerase during the transcription. So you can see how the activator is going to interact with RNA polymerase so that transcription does happen of that target gene. On the other hand, on the right side, you have the repressor, and it is attached to its respective binding site. And what it's done is it's actually physically stopping the RNA polymerase from attaching. So by doing that, it means that no transcription can happen, and that target gene never gets transcribed and therefore will never be translated. So this is one way we can switch it off, is by physically having something there that stops that RNA polymerase from attaching and being able to do transcription. So this is our switched on versus switched off. So as we've said, switched on genes are active genes. They are being transcribed and translated. You are getting their protein product at the end. So switched on genes are being transcribed and are producing mRNA. You've got to remember that the genes that are switched on will be different between cells of different tissues. If they're in different tissues, they need different genes. But you'll also see differences in cells that are in the same tissue when they're at different stages of development. If you have a new kidney cell versus an older kidney cell, they don't necessarily have the same gene switched on because they're doing slightly different things because their development stages are different. Cells under different environmental conditions will be different. So when we say exposed to drugs there, it doesn't necessarily mean illicit drugs. It might mean a medicinal drug, for instance. That is going to create a change in an environmental condition that the cell will interact and respond to. Also, cells that are abnormal, that are doing something that cells don't normally do, like cancer cells, those are going to also potentially have different genes switched on, particularly something like cancer cells, where you're looking at a couple of different types of genes. You're either looking at ones called tumor suppressor genes that normally stop uncontrolled cell growth. So these tumor suppressor genes are really good at making sure that cells don't get out of control and replicate too much. If you get those genes switched off, then suddenly they're not there to stop this uncontrolled cell growth. And so you'll get cells becoming cancerous because they start growing lots and lots when they shouldn't be. So that's one way you can have genes getting switched on off and being a bit different that can lead to cancer or just behaving abnormally. There are other genes that can lead to cancer. It's not just tumor suppressor genes, but that's just one example. So these are your switched on genes. On the other hand, we also have switched off genes. So those genes that are switched on could end up being switched off, and some genes will always be switched off, and you only need the ones that are relevant to what the cell is actually doing to be the ones that are switched on in that cell. So, think about it. If you had the cells in your eyes had their genes switched on that were the ones that made acid in your stomach cells, then your eyes are going to start making acid, and they don't need to do that. It's not part of their job at all. So we make sure that only the genes that need to be switched on in a cell are actually the ones that are switched on. And the way that you control what is on is with those transcription factors. They're going to dictate whether that gene is switched on or off. Okay, so that is how we can control what is going on and what is being expressed in these cells. So we've got another question here. It says the Hox genes are master regulatory genes that influence cells in a particular location of an animal embryo in order to develop structures for that part of the body. In the brine shrimp, Artemia, the expression of Hox genes UBX and SCR results in the growth of either a swimming appendage or a feeding appendage, depending on whether the genes are expressed in cells that are in the mid-region of the body or are near the mouth. These specialized appendages are labeled in the diagram below. So you can see them there in the diagram. Now, you need to describe one way that genes are regulated so that the same genes can produce different appendages when the genes are expressed in different locations in the Artemia embryo. So have a bit of a think about this and see if you can come up with a reason why you might see this difference. Now, there are different answers to this, but you only need to come up with one way because there are multiple ways this could happen. So you can pause the video and write down your answer. Remember, we'll have a video going through this question and its answer, and that video will be linked down below so you can watch it there. So you can pause the video now and go off and try answering it. So now we're going to watch a video on gene regulation. Just before we play it, just be aware that the example towards the end of the video is around the 𝕃𝕚𝕚𝕟𝕂 as in its example, but we don't cover the 𝕃𝕚𝕚𝕟. We'll be covering the 𝕃𝕃𝕚𝕟 instead. So it's good to watch to see how an Operon and transcription factors and all those sorts of things can interact with each other, but the specifics of that as an example is not what we're covering. So just be aware of that.
Have you ever thought about what a disaster it would be if the cells in your eyes started producing the same hydrochloric acid that is made by your stomach cells? Your stomach cells produce hydrochloric acid to help break down food, but you definitely don't want that in your eyes. Thank goodness that doesn't happen. But it's surprising because both your eye cells and your stomach cells contain all of your DNA. Your DNA, all of it, is found in your body cells. But see, the portions that are used need to be regulated somehow. Otherwise, we could end up with something ridiculous, like eye cells producing hydrochloric acid. And that wouldn't just be a waste of resources, that would actually be very difficult to explain to your friends. You want genes to be regulated, controlled. Remember that genes are made up of DNA. DNA is used to give instructions for the production of proteins in the process of protein synthesis. But an important concept is that there needs to be a method of determining which genes will be turned on and which genes will be turned off. This is called gene regulation. And there are many ways that genes are regulated. In your human body cells, you can have proteins that can bind to certain gene regions to increase the rate of transcription for the transcription enzyme RNA polymerase. Or you can have proteins decrease transcription to the point that it may not be transcribed at all. This is a form of gene regulation. Your eye cells don't use the portion of DNA that codes for making hydrochloric acid like your stomach cells do, because there is regulation in your cells to determine which portions of the DNA is used. But we want to shift gears now to talk about a very interesting way of regulating genes that can be sometimes a little challenging to visualize. This particular method is really found in prokaryotes, a few eukaryote exceptions. It's called an operon. An operon is a fancy way of regulating genes, and it's usually made up of a few genes that can involve enzymes. Remember that enzymes are proteins with the ability to break down or build up the substances that they act on. Now let's talk about some key players in an operon so that we can see some gene regulation here. First, RNA polymerase. It's a builder enzyme actually, because RNA polymerase is an enzyme. In biology, a lot of enzymes end in that ASE. RNA polymerase is needed in order to start transcription. Remember that transcription and translation are steps in protein synthesis. Protein synthesis means to make proteins, enzymes in this case. The thing about RNA polymerase, though, it gets a little confusing for RNA polymerase without somewhere to bind. If you watched our DNA replication video, you learned about DNA polymerase and how it had to have a primer to know where to start. Well, RNA polymerase needs a promoter. A promoter is a sequence of DNA where the RNA polymerase can bind. So you would think, okay, you got your RNA polymerase, you attach it to a promoter, and boom, you make your mRNA, which eventually will be used to make a protein. But there's this other sequence of DNA called an operator. The operator is a part of the DNA where something called a repressor can bind. The repressor, if bound to the operator, blocks RNA polymerase, and poor RNA polymerase cannot move forward and no mRNA can be made, therefore no proteins. So let's take a look at our setup here. This is an example called a lac operon. Notice there's a promoter region of the DNA, the operator region of the DNA, and there are 3 genes here that code for enzymes that help in the process of breaking down lactose. Lactose is a sugar. If lactose sugar is around, bacteria want these enzymes to be made so that they can use them to break down the lactose sugar. Then they can metabolize it. Fed bacteria are happy bacteria. Here's the repressor. There's actually a gene here that codes for producing the repressor. See this gene that we call I? It has its own promoter. Well, this gene codes for the production of the repressor. So why do we need this repressor? Well, it's wasteful to make things that you don't need. If there's no lactose, it wouldn't make sense to start making enzymes that work together to break down lactose. It would be a waste. The enzymes would just sit there. So if lactose is not present, then the repressor binds to the operator, and this blocks RNA polymerase. So mRNA cannot be made, and therefore the proteins, the enzymes in this case, cannot be made. But if lactose is around in the environment, something pretty cool happens. The lactose, remember that's the sugar, binds to the repressor. This changes the repressor's conformation. Try as it might, the repressor cannot bind to the operator. RNA polymerase finds its promoter, binds, and transcribes to make the mRNA from the genes on the operon. That mRNA will be used to make enzymes to break down the lactose sugar. And bacteria like to eat, so that makes them pretty happy. We have to say that we think it's pretty impressive to think about all the gene regulation that occurs in cells. And if you find it fascinating, know that there are careers that focus on gene regulation and understanding how genes can be turned on and turned off. Because by doing that, we can gain a better understanding of a variety of diseases that have gene influences in the human body. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
So here we have a slide. Just want to summarize some of the similarities versus differences between eukaryotes and prokaryotes regarding the genetic information. So for starters, there are differences in how many chromosomes they actually have. Prokaryotes will have 1 chromosome. Eukaryotes have multiple, and it varies based on the organism. The chromosome in a prokaryote is circular and doesn't have any ends, so it doesn't have telomeres. On the other hand, in eukaryotes, we have linear chromosomes. So these are sort of straight line chromosomes, and they do have ends, and that means on the ends they have to have telomeres. Prokaryotes have extra DNA in the form of plasmids, and these are much smaller circular bits of DNA. So circular like the chromosome, but much, much smaller. Eukaryotes, on the other hand, don't have plasmids, but you can get DNA from other locations other than the nucleus. Specifically, we're talking about mitochondrial DNA. So there's little bits of DNA in the mitochondria, and for those cells that have chloroplasts, chloroplasts are similar, and they also can have little bits of DNA called chloroplast DNA. So that's sort of where the DNAs can be located in those types of cells. Prokaryotes have nowhere near as much DNA as a eukaryote do. They're way more simple organisms. So a prokaryote is going to have only thousands to millions of bases, depending on the species, whereas a eukaryote, we're talking millions to billions of bases, again, dependent on the species. So it's much more complex, and they have much more genetic information in them. In prokaryotes, they have fewer genes than in eukaryotes. So prokaryotes, we're talking a few thousand genes, whereas in eukaryotes, we're talking tens of thousands of genes. Prokaryotes, the information that they do contain is mostly coding. So what that means is in prokaryotes, their DNA tends to be, for the most part, mostly DNA that will actually code and contains instructions for something. They don't have much non-coding DNA. So they actually have quite a lot of information packed into what they do have. Eukaryotes, on the other hand, have non-coding DNA, or far more non-coding DNA than prokaryotes. So what this means is, think back, eukaryotes have introns, for example, in their genes, whereas prokaryotes don't. Prokaryotes, their genes are purely exons, and so everything that's there actually will get transcribed and kept in their RNA, whereas a eukaryote, they have to go through splicing. You don't have to do splicing with a prokaryote because they don't have introns, because they don't have that non-coding intron DNA. I should add, that is not to say that prokaryotes have no non-coding DNA, just they have a lot less of it compared to eukaryotes. Prokaryotes also don't have a nucleus because they don't have organelles. It's one of their defining features. What that means is the DNA is just free floating in the cell. Eukaryotes, on the other hand, have organelles. They have membrane-bound organelles. It's characteristic of them. And the DNA has to fit into the nucleus. To make sure all that DNA can fit into that nucleus, it is very carefully packed up. And that's done by tightly coiling it around proteins called histones to form something called a nucleosome. That is then condensed into another thing called chromatin, and this all gets packaged as the chromosome into the nucleus. Basically, there's a lot of DNA that has to be packaged very tightly and carefully to fit into a very small space. In terms of controlling the genes that they have, prokaryotes tend to have their genes grouped into functional groups, or they can have them grouped in these functional groups called Operon or Operon regions. So what this means is genes that their protein products will interact with each other. The genes that code for those protein products will also be near each other because their products all interact with each other. Because they're all near each other, they can also have a regulatory region right near those genes that will control all of them as one. So what that will mean is if it's switched off, they're all switched off. If it's switched on, they're all switched on, and that's good because, remember, their protein products will work together. So this is one way that prokaryotes can help regulate their genes, and that's something that we'll be looking at with the 𝒃𝕖𝕐𝑃𝑟𝑜n. Eukaryotes, on the other hand, do not need to do that. They don't have Operon regions, and even if genes code for enzymes or proteins that do similar things, they can be really far apart. They could even be on different chromosomes. They have other ways of controlling gene expression. So they do their own thing, and it's far more complex than what prokaryotes do. So talking about operons in prokaryotes, they are based around genes that code for proteins/enzymes that are in what we call the same biochemical pathway, the same process that they do stuff in. So what you'll have is they will be near each other on the chromosome for the prokaryote. So that grouping is an operon. You can think of operon meaning something like operational region. It doesn't actually mean this, it's just a handy way to remember the term. Okay, so an operon is like a region of things that operate together. It's a very, very simple way that we can group genes, but it's really useful for understanding the basics of gene regulation. And it's also really useful for having simple gene regulation, which is what a prokaryote will want. Eukaryotes are far, far more complex, and there is some research suggesting that they may have some operons, but they're very, very rare if that is the case. And generally speaking, what we say is that prokaryotes have operons and eukaryotes don't. And that is what you'll need to sort of take forward in your thinking. So eukaryotes do not have operons, but prokaryotes do. So you can see here in our picture that we have this section of DNA. You've got a regulatory gene labeled I at the start. that might produce things that can help regulate our operon. But if we look at the operon, we'll see we've got at the beginning a control region, and then we've got structural genes. Those genes, Z, Y, and A, will produce proteins that work together to do something. The control region, you have a promoter and an operator. These are two sections where different things combined to then determine how that and when and if that gene, those three genes rather, will actually be expressed. And we're going to use the trip operon as a way of looking at how this can happen. So let's talk about the trip operon now. It is an operon that is found in E. coli bacteria, and the TRP is a reference to the amino acid tryptophan. And that's what this pathway is all about. It's actually about producing tryptophan, this amino acid. It's an amino acid you can find in your environment, but if there isn't enough in the environment for bacteria, for E. coli specifically, they are actually able to make tryptophan. They have the genes that will produce proteins, that the proteins working together are able to create this new amino acid. Not this new, or they're able to replenish this amino acid if the cell needs it, because there would be none in the environment. Humans can't actually do this in their cells. We don't have the ability to produce tryptophan, so we actually have to get it from our diet. So it's really important that we have food that has the tryptophan amino acid in it, just like all our essential amino acids. We can't produce them, so we have to make sure we consume them. That's why it's important to make sure you're having proteins and a variety of proteins, so you're getting that variety of amino acids. E. coli are able to do this, so they don't have to eat it. They can produce it as long as they have the right precursor molecules that they can then convert into tryptophan. Now, if the trip operon is expressed, so it's turned on, what this means is that the tryptophan levels are low, and so we need to turn on the operon, and we need to then make more tryptophan. And we'll make more tryptophan by, we make these specific proteins, they then work together to make tryptophan. On the other hand, if there is a lot of tryptophan already in the environment, we don't need to do this pathway. If there's a lot of tryptophan, there's no point producing the things that will make tryptophan, because you don't need to make tryptophan anymore because there's already plenty of it. So what you need to understand is that when tryptophan levels are high in the environment, the operon is switched off. And when tryptophan levels are low in the environment, the operon is switched on. So that is the gist of the trip operon. And we're going to watch a video to learn a bit more about it.
Hi everyone, this video is on the tryptophan operon. Tryptophan is an amino acid that's necessary to make proteins, and it's not commonly found in the environment, so bacterial cells often have to synthesize it. So let's look at the components of this operon. There is a gene for the repressor called TRPR, and this gene is always on. It is always expressing the mRNA and always expressing the repressor. This repressor plays an integral role in the operon, but it's not connected to the operon. If we look at the actual operon, we have a promoter and we have an operator, which is a cis-acting site in between the promoter and the genes. And then we have all the genes involved in the operon, and they're all connected together. In this case, we have TRPE, D, C, B, and A. These all code for enzymes that are involved in the biosynthesis of tryptophan. The resting state for this operon is it's on. In other words, the genes are being expressed in the resting state. So, as I said, the TRP repressor is always being expressed. So that means the RNA polymerase can come along and read the gene and produce the mRNA, and then that mRNA codes for a repressor protein. And this repressor protein has a specific shape that makes it unable to bind to the operator region in its current shape. Now, normally, as I said, cells do not have tryptophan in the environment, so they have to make their own. And that means that the RNA polymerase would come along here and bind to the promoter and read all through the genes and produce a polycistronic mRNA that then would code for the different enzymes involved in the biosynthesis of tryptophan. So sure enough, they'll make some tryptophan, and that tryptophan is instantly then used to build proteins because it's an integral amino acid in the synthesis of proteins. So the RNA polymerase would have to keep reading along, making more mRNA, and make more of the biosynthetic enzymes involved in making more of the tryptophan. Now, if all of a sudden tryptophan was available in the environment, why would the cell want to make something that was available for free? When tryptophan is available in the environment, it can actually bind to the tryptophan repressor. This changes the shape of the tryptophan repressor so that it now fits and binds to the operator region. This blocks the RNA polymerase. After it's bound to the promoter, it can no longer have access to the genes. So what we have is the operator being blocked by both the repressor and tryptophan, which in this case is acting as a co-repressor. When the repressor is bound to tryptophan, changes its shape, allows it to bind to that operator. That turns off the expression of the biosynthetic genes responsible for making tryptophan. But as I said, when tryptophan is available in the environment, why would you want to make something that's available for free? If at some point then all of the tryptophan in the environment gets used up to make proteins, and even the tryptophan that is operating as a co-repressor gets used up to make proteins by the cell, this repressor would then revert back to its original shape, which makes it unable to bind to that operator region. This would open up the promoter, and the RNA polymerase would be able to travel from the promoter through to the genes, and again, making an mRNA and making more of the biosynthetic enzymes. So in this way, this is a self-regulating system. When there is tryptophan in the environment, the operator is off because the cell doesn't want to expend energy making something it can get for free. When there is no tryptophan in the environment, the repressor is not able to bind to the operator, and the operon is on, so the genes are able to be expressed and tryptophan can be made for the cell.
So that's a great first introduction to the Tryptophan, and now we're going to again look in a little bit more detail with this. So we're going to watch our second video here.
Two of the most studied operons are the Tryptophan and the 𝕃𝕚𝕚𝕟𝕜𝕤. And what I want to do in this video is focus on the Tryptophan, which is essential for the production of tryptophan. Tryptophan, which you might recognize as an amino acid, often associated with Thanksgiving and turkey dinner. But tryptophan, as all or most amino acids, are essential for creating the polypeptides, the proteins that you use in your body. And so the Tryptophan, and here we're going to be talking about not your body, well, we're going to be talking about something that's in your body. We're going to talk about E. coli. It is an operon that is on the E. coli, that is part of the E. coli genome. And just in this diagram, the way it's drawn, it would be sitting right over here. And just as a reminder, an operon is a combination of a set of genes as well as the regulatory DNA sequences for that set of genes. In particular, you have the promoter, you have the operator right over here. The promoter is where the RNA polymerase binds and would start the transcription process. The operator is where repressor binds. And this is going to be essential for understanding how the Tryptophan works. And so what do these genes actually code for? Well, these genes code for enzymes that are used in the construction of tryptophan. And I'm always amazed that enzymes can be used to construct what are essentially molecules that are much smaller than the enzymes themselves. In fact, the enzymes involved are made up of amino acids, but then they're used to make particular amino acids. And so TRPE, D, C, B, A, they're all, once they are transcribed into mRNA and then translated in the ribosomes, these enzymes are used to create tryptophan for tryptophan biosynthesis. So let's think about how this works. So if we are in a low tryptophan environment, our E. coli, it needs tryptophan. It needs that amino acid as a building block for its proteins. So in that world, it makes sense that in a low tryptophan environment, the RNA polymerase can just latch on to the promoter and begin the transcription process. Transcribe these 5 genes into mRNA, which then can be translated into those enzymes, and then you will have more tryptophan biosynthesis. That makes sense that you want to create tryptophan if you're in an environment that does not have a lot of tryptophan. But what if we did have a lot of tryptophan? Well, if you have a lot of something around, you shouldn't waste energy creating more of it. You have to appreciate that all organisms that are around today are the byproducts of billions of years of evolution, and they've learned to be very careful, or the ones that are selected for tend to be the ones that don't waste resources. And so when you have tryptophan around, you probably don't want this transcription to occur. So it would make sense that maybe tryptophan can act as a co-repressor for a repressor molecule, for a repressor enzyme that would attach to the operator and block the RNA polymerase from transcribing. And that's exactly what happens. So when you're in a high tryptophan environment, and tryptophan obviously does not look like these little yellow quadrilaterals over there, but that's just for our visualization purposes. And neither does RNA polymerase look like that, or neither does the TRP repressor look like that. In fact, I encourage you to web searches and see how they actually look. They're fascinating. But when you have a lot of tryptophan, the tryptophan can act as a co-repressor. It can bind to the TRP repressor, essentially activate it so that it'll change its conformation so that it can then attach to the operator in the Operon. And once it's attached to the operator, well, then the RNA polymerase can no longer move forward with transcription. So as you can see, this is a very valuable feedback loop, or not even necessarily feedback. If you're in an environment with a lot of tryptophan, don't create tryptophan. Or if you just have a lot of tryptophan laying around, don't create more tryptophan. If you don't have tryptophan around, well, then the repressor won't be co-repressed, I guess you can say, and then the tryptophan will actually be created. Now, tryptophan's an interesting thing because the control of transcription isn't the only place where you have some type of a feedback loop or kind of a conditional situation. You can actually have direct feedback inhibition between the proteins. And so this part isn't related to the transcription. But if this is a precursor of tryptophan, it's all very abstract in this diagram. And let's say enzyme 1 turns into precursor 2, enzyme 2 turns into precursor 3, and enzyme 3 turns it into tryptophan. Well, you actually have direct feedback inhibition where tryptophan can then bind or interact with enzyme 1 here. Could interact with enzyme 1. Let me do it in a color you could see. Could interact with enzyme 1 so that it can no longer act as efficiently taking precursor 1 to precursor 2. So this right over here, this is the classic feedback inhibition. Feedback inhibition. The focus of this video, we're talking about operons and gene regulation. But it's important to realize that the regulation of the creation of tryptophan doesn't only occur at the transcription level. And I'm not going to go into this video. It's a slightly more advanced topic, but there's also regulation of tryptophan biosynthesis through a process called attenuation, which doesn't affect the start of transcription, but it affects how things get completed, and it will keep tryptophan from being completely, or the entire process from going to completion. But the ones that are most typically talked about are what we just talked about here, where you have your tryptophan acts as a co-repressor of the TRP repressor, and also the feedback inhibition, which once again is not really about gene regulation, but you can see how the product of this process can go back and inhibit one of the first enzymes.
So hopefully those videos are starting to help you get your head around what's happening with the Operon. There are a couple of links here for some extra videos you're welcome to go and watch, and I'll put the links to them in the description as well, just to help you fully start getting your head around what is happening here. But we'll talk a bit more detail about the Operon, its structure and functionality as we go ahead. So with the Operon structure, you have 5 TRP genes: TRPE, TRPD, TRPC, TRPB, TRPA. Really, it's TRP and then one of the letters from A to E, and they're just listed in reverse order in the gene. So you have these 5 genes. These 5 genes will produce enzymes that work together, one after the other, like a chain reaction. And what their job is to basically, as a group, they work to produce tryptophan. So that is what we have here. So this sequence of genes doesn't produce tryptophan itself. It produces proteins that are enzymes which work together to make tryptophan. So there's a little bit of an extra step in there. Just make sure you're clear on that. The Operon does not itself produce tryptophan. It produces the proteins needed to make tryptophan. So just make sure you're clear on that. Before you have these 5 structural genes, you have 2 special regions. One is a promoter and the other is an operator. Now the promoter, and promoters you see in both eukaryotes and prokaryotes, and we have mentioned them in a previous video, these are regions where RNA polymerase attaches to. It's the RNA polymerase binding site. So it's really important that that comes before the genes you want to transcribe, so that RNA polymerase attaches to the DNA before them so it can transcribe them. As this is an operon in a prokaryote, it has another section after the promoter called an operator. And the operator is basically going to dictate whether or not these genes are operational and they get to be transcribed. If you have a repressor attached to the operator, then the operation is blocked, it is repressed. If it is clear, then it's able to go ahead. Basically, the operator is able to have something bind to it and act as a roadblock to the RNA polymerase. And that will dictate, if it's there, it will stop the RNA polymerase from moving. And if it's absent, the RNA polymerase can move ahead and do transcription. So these are the sort of 3 key regions. The promoter, where the RNA polymerase attaches. The operator, that can have the repressor bound or not there. And then the structural genes. In this case, there are 5 specific genes that make those proteins that will in turn themselves make tryptophan. So this is our structure of our operon. Here we have our 4th exam question. It says the Tryptopan in prokaryotes illustrates the switching off and on of genes. The operator within the Tryptopan is A, is a regulatory gene, B, attaches RNA polymerase, C, codes for the production of an enzyme, or D, is the binding site for the repressor protein. So you can pause the video and go and have a think and answer this question. There will be, again, a video with the answer for this linked in the description, so you can go and check out what the answer is there. Otherwise, pause the video and have a bit of a think about it, and then come back and we'll continue on. So pause the video now. So we've talked about the structure of the Tryptopan. Now let's talk about how it actually regulates things. And we're going to start off sort of talking about basic regulation in a basic way, and then we're going to get more and more in detail as we go along. So in the most basic way of thinking about it, that you have a promoter, an operator, and your structural genes. If there is lots and lots of tryptophan, you do not need, or rather the E. coli does not need, those genes transcribed, because those genes will make more tryptophan. If you already have a lot, you don't need things that will make more. So when there's high levels of tryptophan, that tryptophan that's present will attach to the repressor molecule. And once the tryptophan attaches to the repressor molecule, the repressor is now able to bind with the operator region. And as soon as the repressor binds to the operator region, it becomes a roadblock for RNA polymerase. And that means that RNA polymerase, whilst it can attach to the promoter, it cannot move past the repressor. Therefore, it cannot transcribe those structural genes. So when there's high tryptophan, the tryptophan attaches to the repressor, allowing the repressor to attach to the operator, causing RNA polymerase to be blocked and stopping transcription from happening. On the other hand, if you do not have high levels of tryptophan, then you never have tryptophan attached to the repressor. If you never have tryptophan attached to the repressor, then the repressor is unable to attach to the operator. If the repressor is unable to attach to the operator, then the RNA polymerase won't be blocked. And if RNA polymerase isn't blocked, then it will go ahead and it will do transcription. And so the RNA polymerase will go along and it will transcribe those structural genes. It will then make the proteins that they code for. And once those proteins have been made, they will work together to produce the tryptophan that the cell needs. So this is the difference between what happens at high levels of tryptophan and versus low levels of tryptophan. And it's also how the repressor gets in there and has an impact on whether or not transcription does or doesn't happen. So that TRP repressor is really important, and it's also really important to understand what's happening when there are high levels of tryptophan versus when there's low levels of tryptophan. So, like I was saying, high levels of tryptophan. If the tryptophan is present, it binds to the TRP repressor protein. Once it binds to the repressor protein, it creates a shape change within the protein, within that repressor protein. And once that repressor protein has its shape changed, it ends up becoming active and it ends up becoming a shape that will now actually be able to attach to the DNA. And it does this upstream of those TRP genes. And once it's attached, it's acting, like I said, as that physical block. It's a blockade there. And the RNA polymerase cannot get past it, even if it does attach to the promoter. So this is how, when tryptophan levels are high, you end up with no transcription. That the high tryptophan levels mean that there's tryptophan available to attach and activate that repressor, which means the repressor attaches to the operator and it blocks RNA polymerase from doing anything and doing any transcription there. On the other hand, when you don't have high tryptophan, then there isn't enough tryptophan to attach to the repressor. If there's no tryptophan attaching to the repressor, it never gets activated, which means it's never able to attach to the operator, which means it cannot block RNA polymerase. So RNA polymerase gets a free run at transcribing those structural genes, which is good because they make tryptophan. They make proteins that will make tryptophan, which means you can start bringing that level back up until it's high enough that we end up going back to high levels and you have tryptophan that then attaches to the repressor and it causes it to attach to the operator and stop the process. So you can kind of see there's a bit of a cycle happening here, that if you have low levels, you end up having RNA polymerase do transcription of the TRP genes. They end up producing the proteins. They make tryptophan. As they're making tryptophan, the tryptophan levels increase until you get to the point where now you have high tryptophan levels. Then that tryptophan will bind to the repressor, activate it, the repressor binds to the operator, and now RNA polymerase stops transcribing those genes. So we stop having the proteins being produced, and so we're not continually producing tryptophan by those proteins anymore. So we stop producing, basically, we're able to stop producing tryptophan once we have high levels. So there's a nice bit of negative feedback happening there as well. So you can see here what is happening where you have an inactive repressor. It can't stop the RNA polymerase. So the RNA polymerase goes along, transcribes those genes, the mRNA will go off and it will get translated and you'll produce the proteins. There is another process where you can regulate the Operon, and it's called attenuation. It's a bit of a tricky process, so we're going to work through it a little bit slowly. Basically, when tryptophan levels are high, there is another way that you can stop it being, the protein products being produced. It doesn't entirely stop transcription, where, when you have the repressor protein bound to the DNA, no transcription happens. With attenuation, you will still get some transcription, and in fact you'll get a little bit of translation, but it never gets completed properly, and so you never actually end up with the final protein products at all. And you can see sort of a summary here that the RNA polymerase starts transcribing, but you have high tryptophan levels, and those high tryptophan levels mean that the mRNA gets actually made quite quickly, and forms these termination structures. The ribosome that's translating reaches a termination structure, and that causes the mRNA to detach, where, if you actually want to make the protein products, you don't want that happening. Once that detaches, the RNA polymerase detaches from the DNA strand itself and it stops transcribing. And so you end up with only this little bit of mRNA being made, and you end up with a little bit of it being translated, but you don't get anything translated into the final gene products, which means you never end up with your final proteins, which means you never end up with the things that make tryptophan. So it's a slightly different process for regulating the 𝕃𝕚𝕚𝕟𝕜𝕤 in a bit more detail. All right. So it's a bit of a funky process. One thing you have to keep clear in your head is that this is a prokaryotic cell. It doesn't have organelles. The DNA is not in a nucleus. So because the DNA is just in the cell around things like ribosomes, it means that the ribosomes don't actually have to wait for RNA polymerase to finish transcribing. Ribosomes can actually start translating the mRNA as it's coming off the DNA with the RNA polymerase transcribing it. And that's just because there's no organelles and everything's in the same space. So you can see in the bottom right corner there, you have mRNA being transcribed and it's coming straight off that DNA into a ribosome. And so it's happening one after the other, like almost together. That's sort of key for how attenuation can happen. So in E. coli, the ribosome attaches to the mRNA as it's still being transcribed off the 𝕃𝕚𝕚𝕟. There's a little bit at the very start called the leader that we hadn't talked about before. And this leader section of the mRNA, as it gets to the ribosome, and it's being read and translated into a polypeptide, what's really interesting is in that leader section are two codons that require tryptophan tRNA molecules. Now, if you've got high levels of tryptophan, then the ribosome does not have to wait long at all for those tRNA molecules with tryptophan to appear. So the ribosome, if there's high levels of tryptophan, the ribosome can fill in those two codons very quickly, and it moves on very quickly. If there's low tryptophan, the ribosome gets stuck and it stops, and it has to wait for this tryptophan carrying tRNA, and it can take a while because you don't have high levels of tryptophan. So what happens is the ribosome slows down and it gets held up. It's kind of in a bit of a jam there. What this means is that when you have high tryptophan levels, the translation happens quicker. When you have low tryptophan levels, the translation happens slower. You're probably thinking, so what does that have to do with anything? Well, depending on how quick or fast that translation is happening, has an impact on how quickly the mRNA is moving through the ribosome. And how quickly the mRNA moves through the ribosome can allow one of two things to happen with that mRNA that's following on behind. So after the leader section, there's an attenuator section. So you can have a look down the bottom and you can see between sort of the operator and the first structural gene, there's this bit of a region in there where you have the leader and then the attenuator. As you transcribe that attenuator, the RNA it forms, or the mRNA it forms, can stick to itself. And it can stick to itself in two different ways. Either this region 2 and region 3 can stick to each other, or region 3 and region 4 can stick to each other. How fast that translation happens, remember, I said has an impact on how quickly the mRNA moves through the ribosome. If you move the mRNA through the ribosome quickly, that causes 3 and 4 to stick together. If, on the other hand, you move slower, it causes 2 and 3 to stick together. And we'll see that on the next slide. But basically, what will happen is if you translate fast, as this mRNA changes shape, as it's moving fast, it causes something called a terminator hairpin. And a hairpin is just a really sharp loop. Fast translation causes a terminator hairpin, whereas slow translation causes an anti-terminator or a non-terminating hairpin. That's the kind you want if you want to keep transcribing. And you only get that with slow translation, which only happens when there's low tryptophan. Remember, when there's high tryptophan, the ribosome is able to translate much faster. And you can see when it translates fast, it causes the terminator hairpin, the kind that will cause transcription and translation to stop. So if you have your terminator hairpin there, the RNA polymerase detaches and the ribosome also hits that and detaches. And so it stops transcription and translation if you have the terminator hairpin from faster translation. But if you have the slower translation and you get the anti-terminator hairpin, then that actually stops the terminator one from ever forming, and it means that transcription continues. This is what we're talking about. So have a look at where it says high tryptophan levels. As you have your RNA polymerase going along and it's making its mRNA, the ribosome gets through that leader really quickly. As it gets through the leader really quickly, it ends up actually covering up section 2. Because section 2 gets covered by the ribosome, section 3 can't attach to it. So instead, section 3 attaches to section 4. And so what we have is we have 3 and 4 attached, and they make a terminator hairpin. Now, as soon as the ribosome gets there, it goes, no, can't do this, and it detaches. And the RNA polymerase, as that has also been formed, the RNA polymerase will also detach. What this means is before you ever get to transcribing the structural genes, you've created something in the mRNA that causes RNA polymerase to detach and stop transcribing, and also causes the ribosome to detach and stop translating. So with these high tryptophan levels, because things moved quickly, it basically all falls apart. On the other hand, if you have those low tryptophan levels, it means that the ribosome moves more slowly, which means region 2 doesn't get covered up, and it can now attach to region 3 as soon as region 3 is made. Well, when 2 and 3 attach to each other, they don't make the terminator hairpin, they make the anti-terminator hairpin. So because they've made that one, and now 2 and 3 are attached, so 3 is not going to attach to 4, so you're not going to have that problem with the terminator hairpin. They've made a happy little anti-terminator hairpin, so RNA polymerase doesn't affect it. It is able to continue transcribing, and when that anti-terminator hairpin gets to the ribosome's fine with it, and it keeps translating, and it's all good. So you can see when there's high tryptophan, we have this process where the high tryptophan causes this initial translation to happen too quickly. It causes these terminator hairpins to form, which cause everything to detach and stop transcription and translation, which is good because you have high tryptophan and it doesn't need to happen anyway. On the other hand, when you have the low tryptophan, things move more slowly and you end up with a different type of hairpin called a non-terminator hairpin forming, which this one means that transcription and subsequently translation can continue on, which is good because you need them to make those protein products so that you can make more tryptophan because you don't have enough of it. This whole scenario with these terminator hairpins and these anti-terminator hairpins, that is attenuation. And that is a different way the TRIP operon can be regulated. So it's a little bit of a tricky one to get your head around, but just understand that when you have high tryptophan, the ribosome moves quickly, you end up with terminator hairpins that causes transcription and translation of the operon to stop. On the other hand, if you have low tryptophan, the ribosome moves more slowly, you have a non-terminator hairpin forming, and transcription and translation are able to continue. That is sort of the key that you need to take away when it comes to attenuation here. To help you give you a bit more of an understanding, we'll watch a video. So we've got a video here on attenuation, and hopefully we're able to show you what is happening in action.
The TRIP operon in E. coli contains 5 structural genes corresponding to enzymes that convert 𝗂𝕗𝕗 into tryptophan. The TRPE gene contains a 5' untranslated region, or UTR, that plays an important role in the regulatory mechanism called attenuation. The 5' UTR contains 4 regions. Region 1 encodes a short peptide containing tryptophan amino acids. When tryptophan levels are high, region 3 pairs with region 4. This structure terminates or attenuates transcription. When tryptophan levels are low, region 2 pairs with region 3. This structure does not terminate transcription. Let's examine how these alternate structures arise. RNA polymerase begins transcribing the DNA. Closely following RNA polymerase, a ribosome begins to translate region 1. When tryptophan is abundant, the ribosome doesn't slow down at the tryptophan codons. By the time region 3 is transcribed, the ribosome has already moved past the tryptophan codons to partly cover region 2. Because region 3 is prevented from pairing with region 2, it pairs with region 4 instead to produce the attenuator, which terminates transcription. The structural genes are not transcribed, and therefore no additional tryptophan is synthesized. When tryptophan is scarce, the ribosome stalls at the tryptophan codons. Therefore, region 2 is not covered when region 3 is transcribed, allowing regions 2 and 3 to form a hairpin that does not terminate transcription. Because region 3 is paired with region 2, it cannot pair with region 4 and form an attenuator. Transcription continues. RNA polymerase transcribes the structural genes, which are translated into enzymes, which in turn synthesize more tryptophan. You can mimic the effect of tryptophan levels by determining how long the ribosome pauses at the tryp codons.
We've talked about how you can regulate the 𝗃𝕚𝕚𝕟𝕜𝕤, but I haven't spent much time talking about what it actually does. So the 𝗃𝕚𝕚𝕟 has 5 structural genes, and when you have it switched on, it goes through, it transcribes, and subsequently translates those 5 genes. And you end up with 5 different proteins. You end up with one produced by 𝗃𝕃, one by 𝗃𝐷, one by 𝗃𝐶, one by 𝗃𝐵, and one by 𝗃𝐴. If you're like, I'll never remember them, you don't need to stress too much about remembering them, but it's also just A, B, C, D, and E, but in reverse order. So it's not too hard to remember. Now, I've given you the names of the proteins / they're actually enzymes that those different genes code for. You do not need to remember the names of them at all, but that's there in case you're curious about them. Now, these 5 proteins / enzymes, they work together. And you'll have your first protein will take the first precursor molecule, and it will do whatever it does to it, and it will produce a secondary intermediate sort of molecule. That molecule then goes on to the next one, and it does its thing to it, producing another intermediate. That goes on to the third, again, repeat the process, another intermediate's produced, that goes on to the 4th one, and so on and so forth, until it gets to the last enzyme. And that last enzyme gets it. What it's going to do is it actually takes whatever the intermediate was, the second to last intermediate was, and it's going to turn it into tryptophan. So you basically have this long process of each enzyme doing something different to the molecule until you get to the final one where it just needs that one last thing done to it, and now it is a tryptophan molecule. So overall, the things that get made from the 𝗃𝕚𝕚𝕟 are proteins / enzymes. It's the type of protein they are, as they're an enzyme. You end up with these 5 enzymes that they work together to take these other molecules in the cell and turn them into tryptophan and create tryptophan for the bacterial cell. If you have high levels of tryptophan, this will be stopped, and that is your negative feedback. Once the tryptophan levels increase enough, they activate the 𝗃𝕚𝕚 protein and they switch off the 𝗃𝕚𝕚𝕟, stopping these proteins being made, which means they stop making more tryptophan. So there's a bit of negative feedback for how it all gets controlled there as well. So that is what the 𝗃𝕚𝕚𝕟's genes actually do. And that is actually how it works. We're going to take time to do another exam question. We've got 2 here. And you need this first bit of information to help answer both questions. It says structural genes can be switched off and turned on by transcriptional factors expressed by regulatory genes. In prokaryotes, a group of genes associated with the production of tryptophan is grouped together in a single operon called the 𝗃𝕚𝕚𝕚. The diagram below shows the positions of the genes on the prokaryote chromosome. Question 5. Transcription of the structural genes within the 𝗃𝕚𝕚𝕚 will occur when A repressor molecule is attached to the operator, B, RNA polymerase is attached to the promoter, C, tryptophan is present in the prokaryotic cell, or D, transcription of the TRPA gene is optimal. Question 6 says, transcription of the structural genes within the TRP operon results in the production of molecules of A, the transcription factor, B, a repressor protein, C, tryptophan, D, mRNA. So again, I will have a video explaining the answers to these two questions linked in the description below, and you can pause the video and take a moment to answer each of those questions and then go watch the answer in its own video. So you can pause the video now. To help get your head around what the TRP operon is doing, I have an activity for you. And it's relatively simple. Just using something like PowerPoint or other animation software that you have and can work, I want you to create 2 animations that show how the TRP operon actually is regulated. One of those animations should show what happens when tryptophan levels are high, and another animation should show what happens when the tryptophan levels are low. If you want to, you can also create a third animation that shows how attenuation actually affects the TRP operon. This is a trickier process, but if you think you're up to it, by all means give it a go. What you can do when you're done is you can submit that to me as well, either via Crest Connect or via email. So this is what I want to leave you with and what I want you to go off and do. Otherwise, this brings us to the end of our PowerPoint. If there is anything you did not understand, you can always go back and rewatch that section or reach out to your teacher for further clarification. Otherwise, I will see you in the next video. Bye for now.